Welcome dear students. Our today's topic is the biological oxidation. You are already much familiar with the oxidation and reduction terms. Oxidation means the loss of electrons and reduction means the gain of electrons. In the biological systems, the redox processes they also take place and they are called as the biological oxidation. So the biological oxidation is basically the cascade of the redox reactions that takes place in the biological systems. And there are two main events involved in the biological oxidation. First is the electron transport chain ETC, which is also called as the electron transport system, and the next one is the oxidative phosphorylation. So ETC and oxidative phosphorylation, these are the two major events of the biological oxidation. In this lecture, we are going to discuss in detail what is the electron transport chain. Electron transport chain, that is the final step of the cellular respiration, and it is also known as I have already mentioned that it is also known as the electron transport system, where this electron transport chain process takes place. It takes place in the inner membrane of the mitochondria. Mitochondria is the powerhouse of the cell. It is an organelle which has two membranes. There is an inner membrane, there is outer membrane. The matrix present within the inner membrane is called as mitochondrial matrix, and the space which is present between the two membranes, that is called as the intermembrane space. So this electron transport chain, this complete process takes place in the inner membrane of the mitochondria. And basically, electron transport chain is a series of the protein complexes in which the electrons they travel from low redox potential to high redox potential, ultimately producing the energy in the form of the ATPs. When the electrons they move from low inner redox potential to high redox potential, some of the energy is released and most of that energy is dissipated in the form of the heat, but some of the energy is used to create a proton gradient across this inner mitochondrial membrane, and this proton gradient will activate the ATP synthase to produce the energy in the form of ATPs. Now let us discuss in detail what is meant by this electron transport chain. My dear students, this diagram in front of you that shows the inner membrane of the mitochondria and the matrix that will be present inner side to this membrane, that will be called as the mitochondrial matrix. So this portion of the whiteboard is actually mitochondrial matrix. This diagram is of the inner membrane of mitochondria, and this portion of the whiteboard where I have written these complexes, that will show the intermembrane space. Now there are four protein complexes involved in electron transport chain. Complex 1 is also called as NADH dehydrogenase. It consists of a flavoprotein, flavin mononucleotide, and 8 clusters of the iron and sulfur. The complex 2, which is directly related to the Krebs cycle, it is also called as the succinate dehydrogenase, and it consists of the flavoprotein, that is flavin adenine dinucleotide, and again the iron sulfur clusters. The complex 3 is also called as CoQH2 cytochrome C oxidoreductase, and this consists of the cytochrome C1, two iron sulfur clusters, and cytochrome B. And the complex 4 is also called as the cytochrome C oxidase, which consists of the cytochrome A and cytochrome A3. So these are basically the four protein complexes which will be involved in the transport of the electrons from low redox potential towards the high redox potential. I have also mentioned the redox potential of different substances. NADH has the lowest redox potential of minus 0.32 volt, FMN minus 0.12 volt, coenzyme Q plus 0.04 volt, and then there is cytochrome B plus 0.07, cytochrome C1 plus 0.23, cytochrome C plus 0.25, cytochrome A plus 0.29, cytochrome A3 plus 0.55, and the highest redox potential belongs to the molecular oxygen, and that is plus 0.82 volt. Now, my dear students, first of all, these NADH molecules and these NADH molecules, they are basically derived from the first three steps of the cellular respiration: glycolysis, pyruvate oxidation, and Krebs cycle. So, glycolysis, pyruvate oxidation, and Krebs cycle produces the NADH. This NADH will be oxidized to NAD and protons, and it will release two electrons. These two electrons will pass through this complex one, and they will move to the coenzyme Q. These two electrons will be first given to FMN, then they are transferred to 8 iron sulfur clusters, and finally they move to coenzyme Q. In this complex one, the 4 protons are also pumped from the mitochondrial matrix to the intermembrane space through this inner membrane of the mitochondria, and movement of these 4 protons that will produce a proton gradient across this inner mitochondrial membrane, and this proton gradient is very important to activate the ATP synthase for production of the chemical energy in the form of ATPs. My dear students, in complex 2, the flavin adenine dinucleotide in its fully reduced form that will be oxidized to its fully oxidized form. The 2 electrons released, they will pass through iron sulfur cluster and they will move to coenzyme Q, which is an electron carrier. Now, electrons from this coenzyme Q, then they will move to this cytochrome B of the complex 3. But listen very carefully, in the complex 1, there was pumping of the protons, while in the complex 2, you can see there is no proton pumping involved. So basically, the complex 2 is involved in the least energy production, because energy will be produced at the step where there will be proton pumping through this inner mitochondrial membrane. My dear students, when the coenzyme Q will lose 2 electrons, it will be oxidized, and these 2 electrons will move to cytochrome B. Cytochrome B will accept electrons and that will be reduced. So in this complete electron transport chain, the substance that will lose electron, it will be oxidized, just like in the first step NADH was oxidized and FMN was reduced. Then FMN transferred electron to iron sulfur cluster, it is oxidized and this one gets reduced. So this is a cascade of the redox processes that take place in this electron transport chain. Then the electron moves to cytochrome B, and from cytochrome B to 2 clusters of the iron and sulfur, then to cytochrome C1. And from cytochrome C1, the electron move to cytochrome C. In this complex 3, again there will be pumping of the 4 protons across the inner mitochondrial membrane, and again a proton gradient. Proton gradient means the difference in the concentration of proton between the matrix and intermembrane space that will be created, and it will help the ATP synthase to produce the energy in the form of ATPs. Then the cytochrome C, it gives electrons to its final destination, that is the molecular oxygen, but electron pass through the cytochrome A and cytochrome A3 of this complex 4, and again it will be involved in the pumping of the 2 protons. So here again the energy will be produced, but that energy will be somewhat lesser as compared to the complex 3 and complex 1. So the maximum energy is produced in complex 1 and complex 3 due to the pumping of the maximum number of the protons. And my dear students, in the complex 2, the minimum energy is produced as there is no pumping of the proton. And in complex 4, the energy is produced, but that energy will be less as compared to the energy produced in complex 1 and complex 3. Finally, the electrons will move to molecular oxygen and it will be reduced to water. And the molecular oxygen has highest redox potential. So the electrons, they have traveled from lowest redox potential of NADH to highest redox potential of the O2. And during this traveling of the electrons from the lowest redox potential to highest redox potential, the energy is released, which is used in pumping of the proton across the inner mitochondrial membrane. And this pumping of the proton will create a proton gradient across this inner mitochondrial membrane, and this proton gradient then that will activate the ATP synthase and it will produce energy in the form of the ATPs. So the whole purpose of the electron transport chain is to move the electron from NADH to oxygen and reduce oxygen to water. And the second main purpose is again to produce the electrochemical gradient in the form of the proton gradient across the inner mitochondrial membrane. So that was all about the electron transport chain. And keep one last point in your mind, that electron transport chain is the only step in the complete cellular respiration where the molecular oxygen is directly used. In no other step of the cellular respiration, the molecular oxygen is directly involved. So it was all about the ETC. I hope so that this lecture will be very understandable for you people. In case you find any problem, you can ask in the comment section. I will definitely answer that. Till then, take care.